Hello everyone, welcome to BioLearn with Harshani YouTube channel. My name is Harshani Nadika, so I really enjoy molecular biology, biotechnology, genetics and bioinformatics. If I say about my academic background, I have a bachelor degree especially in molecular biology and plant biotechnology from University of Kelaniya in Sri Lanka. And I am currently doing my master's in molecular biotechnology that is specialized in system biology at the University of Högdö in Sweden. So I started this channel to make these topics simple and easy to understand. Here I will share short lessons, clear explanations and study support for high school students, university students and anyone interested in life science. If you like science and want to learn in a simple way, you are in the right place. So let's get started with this topic, Introduction to Molecular Biology. Let's see what is molecular biology. Molecular biology is the branch of biology that studies biological processes at the molecular level, focusing on the structure, function and interactions of DNA, RNA and proteins and how these molecules regulate cellular activities. Molecular biology is built on three pillars. Those are the genetics, biochemistry and cell biology. Genetics explains how genetic information is stored and inherited, and biochemistry studies the molecules and chemical reactions that make life possible. And cell biology focuses on the structure and function of cells where all molecular processes take place. So together these three fields help us understand life at the molecular level. In 1938, the term of molecular biology was introduced by Warren Weaver, and he and his team proposed using the rigorous physical and experimental tools of the physical sciences to analyze biological structures in biology, biochemistry, cell biology, and genetics. Let's see historical landmarks in molecular biology. First, early discoveries. So in 1928, Frederick Griffith discovered the transforming principle in bacterium pneumococcus, now known as Streptococcus pneumoniae, demonstrating genetic transfer. In 1934, Max Schellinger purified bacteriophages, proving for the first time they are composed purely of DNA and proteins. In 1941, Beadle and Tatum established the classic one gene one enzyme hypothesis to genetic studies of the fungus Neurospora. In 1944, Avery, MacLeod and McCarty proved conclusively that the physical agent of Griffith's transforming principle was DNA. In 1950, Erwin Chargaff formulated Chargaff rules, proving that in DNA, the ratio of adenine to thymine and cytosine to guanine is always 1:1. In 1953, James Watson and Francis Crick proposed the double helical model of DNA based on Maurice Wilkins and Rosalind Franklin. So Watson, Crick and Wilkins were awarded Nobel Prize in 1962. In 1957, Messerson and Stahl confirmed Watson and Crick's semi-conservative model of DNA replication. Here the DNA replicates semi-conservatively, meaning each new helix retains one old parental strand. In 1958, Arthur Kornberg successfully isolated DNA polymerase from E. coli, earning a Nobel Prize in 1959. In 1958, Francis Crick formulated the central dogma of molecular biology, establishing that genetic information flows strictly one way, from DNA to RNA, RNA to protein. Next one is cracking the genetic code. In 1961, Nirenberg and Mette cracked the genetic code present on mRNA. In 1961, Jacob and Monod introduced the operand concept using the lac operand, demonstrating how cells actively regulate gene expression based on environmental cues. In 1966, Francis Crick proposed the wobble hypothesis, explaining how a single tRNA anticodon can recognize multiple degenerate codons on mRNA strand. Hamilton, Smith, Wilcox, and Cayley invented the first restriction enzyme in 1970. They isolated this first restriction endonuclease from bacterium Haemophilus influenzae. These enzymes act as molecular scissors, allowing DNA to be cut at precise predictable nucleotide sequences. Then in 1973, Herbert Boyer and Stanley Cohen invented the recombinant DNA technology. They injected that recombinant plasmid into E. coli cells, and their foundational publications established the structural blueprint for modern genetic engineering. The following timeline highlights the major breakthroughs that have shaped modern molecular biology. Those are the sequencing revolution and PCR revolution. In 1977, Maxim, Gilbert, and Colson described DNA sequencing techniques. Frederick Sanger and his team developed Sanger sequencing method that is also known as the chain termination method. Fred Sanger's team sequenced the very first complete genome of a bacteriophage, and he earned his second Nobel Prize in 1980. In 1985, Kary-Mullis invented polymerase chain reaction that provides a way to rapidly amplify specific DNA sequences. This revolutionary tool earned him the Nobel Prize in Chemistry in 1993. As modern landmarks in molecular biology, in 2003, the complete human genome was mapped, enabling major advances in molecular biology, disease research, and genomic medicine. In 2012, CRISPR-Cas9 gene editing system was invented. So Jennifer Doudna, Charpentier, and their colleagues adapted a bacterial immune system into a simple, programmable tool for genome editing. So this breakthrough allows scientists to precisely cut and modify DNA in many organisms. In 2018, scientists discovered single-cell RNA sequencing method. So the development of single-cell transcriptomics empowers scientists to track gene expression profiles by cell. This dynamic technology breaks tissue averages, mapping precise cellular atlases and disease pathways. In 2020, scientists found COVID-19 mRNA vaccines. In response to global threat, lipid nanoparticle formulated mRNA vaccines are engineered and approved within a year. This validation establishes synthetic mRNA as a versatile clinical vehicle for future cancer vaccines and therapies. Molecular biology can be classified into two: classical molecular biology and new molecular biology. Classical molecular biology focuses primarily on fundamental processes such as DNA replication, transcription, and translation. And new molecular biology integrates high-throughput technologies, system biology, and synthetic biology. Molecular biology is expanding rapidly, and it has given birth to various fields such as bioinformatics, genomics, and proteomics. Molecular biology, that is the foundational layer. It observes the specific interactions between individual macromolecules, DNA, RNA, and proteins inside the cells. Bioinformatics, that is the computational engine. It processes complex biological data generated by modern high-throughput assays using statistics, programming, and mathematics. Genomics, that scales molecular analysis upward from individual genes to the entire genome. Proteomics studies the entire complement of active proteins expressed by an organism at any given time. So these three interconnected fields together form a unified framework that drives modern biological research and discovery. If you found this video is interesting, press the subscribe button, like the video, and share. Leave your comments in comment section. See you with a new video. Thank you for watching.